Hi. So, in Advanced Bioinformatics, today's lecture, we are going to study core data retrieval tools and platforms. There are three major platforms that are studied, that are used for data retrieval: Entries, SRS, and DBGET, database get. These three majorly difference in who is actually controlling them and how many databases these are integrating. So, Entries is developed by NCBI. This is text and sequence based searching platform, and it integrates over 40 databases. And by this, it includes PubMed for literature and GenBank for sequencing. So at the same spot, you have already published data, and at the same spot, you have access to various sequences from worldwide. SRS, which is Sequence Retrieval System, is controlled and governed by EBI, and it is widely used integration platform, allowing users to query multiple biological databases simultaneously. So there are multiple biological databases obtained from multiple resources, and they are simultaneously at the same time being searched through at SRS, and it integrates over 80 plus databases at the same spot. DBGET, database get, is managed by Kyoto University and the University of Tokyo. It is a database integration system highly linked to metabolic and structural pathways. So this database get is actually focusing on only metabolic and structural pathways overall, and it has almost 20 plus databases integrated into it. There is one term, this platform XSpaCy, which is considered a retrieval tool, data retrieval tool, but it is not exactly a data retrieval tool. In fact, it is a bioinformatics portal. And by this word portal, we mean that it provides access to protein-related databases, sequence analysis tools, proteomic resources, all the protein resources and protein analysis tools. So at the same spot, we have databases and analysis tools in XSpaCy simultaneously at the same time. So XSpaCy does perform data retrieval, but its main purpose is protein analysis and access to protein resources. So you retrieve data and you analyze it, unlike entries or SRS, which are only for retrieval. Software tools which are used for data retrieval include sequence databases. Three major categories are in software tools for data retrieval, which include sequence databases, genome browsers, and proteomic databases. Sequence databases include only those databases which directly relate to sequences, either of protein sequences or nucleotide sequences. Like NCBI, GenBank is only for nucleotide sequences. Ensemble is for vertebrates and model organisms genomes, so nucleotide sequences. UniProt is deals in protein sequences and functional information, and database of Japan and EMBL EBI international nucleotide sequence databases. So genome browsers are those browsers which with the help of which we actually visualize various kinds of genomes, and we can compare them and explore their genomic sequencing data and visualize where the problem or where the mutations can arise. So University of California genome browser, UCSC genome browser, visualizes genome annotations and tracks. Ensemble genome browser is used for comparative genomics, and IGV, which is integrative genomics viewer, explores large scale sequencing data like binary data and variant calling formats. Proteomic databases, which are protein data bank. In proteomic databases, we have three major categories for software tools for data retrieval, which include protein data bank, dealing in 3D structure of all the proteins. PFAM is protein families and domains. So we have various groups and families of proteins, and then we can study their domains grouped in like various function domains based on their foldings and their reactive sites. So string is another data retrieval software which deals in protein-protein interaction networks. Next, we have software for data analysis. previously we were discussing data retrieval softwares. Now we are going to study data analysis softwares. For in this we have three major categories: genomic data analysis, proteomic data analysis, and functional pathway analysis. Genomic data analysis again deals only in nucleic acid uh, based data analysis. Data analysis involves one major thing, which is comparison of one sequence in hand to all the other sequences that are present, that are being searched for worldwide. So BLAST is the sequence similarity search. Similarity we discussed in earlier lecture is means that we uh, uh, compare it to other sequence, nucleotide by nucleotide, to find how many sequences, how many nucleotides are aligned and similar in those sequences. This is done by BLAST. MAFT and Clustal Omega is another tool for data analysis, which is used for multiple sequence alignment. Like at the same time, instead of only two sequence similarity searches, you use multiple sequences and align them. Alignment means you compare them to each other. Bowtie is short read sequence alignment, so you read shorter sequences rather than full length sequences. And genome analysis toolkit is for variant calling and genomic data processing, that is very useful in mutation analysis. Then we come towards proteomic data analysis, which includes mascot, protein identification from mass spectrometric data. So those proteins which are being analyzed, which are being studied using mass spectrometry, uh, are identified using mascot. Cytoscope is another software for data analysis, and it involves network analysis of protein interactions. So how proteins interact with each other and how that interaction affects a full cellular environment is studied under cytoscape. Interproscan is used to predict protein domains and functions, which domains are active, which domains are hidden, and how this orientation of protein domains affects their functions is studied under Interproscan. Swissprot is another software and important software for manually reviewing high quality protein sequences and annotation databases. Then we have pathway analysis, functional and pathway analysis for full metabolic pathways. For this we have KEGG for pathway mapping, Reactome for functional networks of proteins and genes, and then we have gene ontology, which is gene functional annotation and enrichment analysis. So by gene enrichment analysis and functional analysis, we mean that we study that whether a predefined set of genes is significantly overrepresented in a large data set uh, after being upregulated or downregulated. So this is gene ontology, and that is all about softwares used in data analysis and data retrieval.